Welcome everyone, and thank you so much for joining us today for Design an Effective Chemical Characterization Study for your device. Our presenters today are Dr. Philip Ferko, Senior Scientist with EAG Laboratories, Dr. Melissa Batting, Senior Managing Scientist with Exponent, and Dr. Megan Arnold, Senior Scientist with Exponent. Our moderator today is Dr. Sahil Bana, Regional Sales Manager with EAG, and I'm Melissa Campbell with Marketing for EAG. Just a couple housekeeping items before we get started. All of you are muted. However, if you have questions during the presentations, we would love to have them. Please put them in the control panel or on your app in the questions section. And if time permits, we will answer these questions during the panel discussions. And if we run out of time, we will follow up with you and your questions in the coming week. At the end of the event, a survey will pop up on your screen, and we would love to have your feedback. And now I will turn it over to Dr. Bana.
Thank you, Melissa, and hello, everyone. I'm Sahil Bana. I'm one of the sales managers at EAG, responsible for our medical device clients, and I'm really happy you guys joined us today. We're excited to partner with Exponent on this event. Exponent's been a trusted partner to EAG over the last several years, and we often team up with them to help our clients design effective chemical characterization studies. We get a lot of questions around best approaches and best practices to chemical characterization, so we decided to put this webinar together to share information and our experiences that we hope will be helpful to you. We understand the importance of making it through a successful study, and the Exponent and EAG team worked very closely throughout this process to help ensure this. So the layout for today's event is going to be split into two sections. We'll start with how to plan a study with presentations from Dr. Ferko and Dr. Batting, followed by a Q&A session for this portion. And then the second part will focus on study completion and results with Dr. Ferko and Dr. Arnold, followed by another Q&A session. So please make sure to send your questions into the control panel as you have them. And again, thanks for everybody for joining. And I'm going to turn it over to Dr. Ferko to get the presentation started.
Thank you, Dr. Bana. So I'm going to first be talking about the topic of initiating a 10993 Part 18 chemical characterization study. Before we get into that, though, I'd just like to give a little bit of my background. So I'm a team lead senior scientist with Eurofins EAG Laboratories, and my work is in supporting chemical characterization of medical devices. So this involves method development, process optimization, a lot of project support, especially on projects that may present technical challenges on the analytical chemistry portion. So the techniques I'm most familiar with are LCMS, FTIR, and Raman spectroscopies, as well as SEM-EDS. Previous work has been in pharmaceutical deformulations and also quality control. So we kind of understand the broader, sort of a production-oriented section, as well as some of the related pharmaceutical work. So before diving into the details, I'd just like to go over an outline of what I'm going to be covering in this first talk. Really going to be answering the questions: who should I involve in starting a chemical characterization study? And what might I need to start a study? In terms of the involvement, this image on the right really, I think, captures this idea. So the three parties that are really involved in a chemical characterization study are the study sponsor or product manager, the analytical team, as well as the toxicological team. And all three of these groups really need to be working together for an effective study that's going to be on time, on budget, and result in a TRA, or toxicological risk assessment, that's going to support that device. So this triad of parties is something I'm going to be touching on again and again, and I think you'll see it's a pretty common theme throughout this entire webinar. So I'm going to talk about some items that you might need or want to have to start a study as well. Those will include the non-disclosure agreement, quote, sample submission form, example device, feasibility study, and the protocol, which will be kind of the results of everything. And this will be the item that will then initiate the next step, which is the actual execution of that study. So start with the non-disclosure agreement. So I'm sure many of you are familiar with these already. It's a standard practice, and the purpose is to protect data and intellectual property. Why it's important for a Part 18 study, though, is that it facilitates a free exchange of information for a successful study. It's establishing that baseline trust that your data and intellectual property will be protected, and it really lays the groundwork for effective communication. Why is that important? It's important because the device context matters. You as the study sponsor have access or are privy to a lot of information that the analytical team or the toxicologist may not have. And so being able to share that information freely and thoroughly is going to help to support a successful TRA. The next item is going to be a quote. This will typically come after some preliminary discussions on scoping out the work. I think there is a tendency to see the quote as sort of just a price tag or a necessary piece of paperwork to complete the study, but it's really actually pretty important for scoping out the work because it's going to describe what work's actually going to be completed. And this is the first document that'll help us ensure that the study's going to meet the needs, right? So will it meet the needs of the study sponsor, but also will it meet the needs of the toxicologist? If your toxicologist is already involved, sharing this document with them can help him or her to determine whether or not the work will be sufficient. There may be instances where the standard approach may not be sufficient, and this can be based on the toxicologist's experience or maybe your awareness about your device. And this is the first point where that can be caught and then addressed. So it's really important. It's really defining the scope of the work, and it's starting that discussion and prep of that protocol and study. So case study for this, the quotes being useful. We've run into situations where we've had ion chromatography requested by the toxicologist. So this is very helpful because it can provide information on structure, not just the elements that are present. So ICP-MS may catch elements that are present, but the difference between, say, sulfur versus sulfate or phosphorus versus phosphate can be important. It's particularly useful for surfactant counter ions. And so a toxicologist that was involved in a project looked at the quote and said that they wanted to also see ion chromatography as well. So at that point, it could be accounted for in the cost. But the other part was it tipped off the lab to make sure that in the study design that there would be sufficient sample to do the regular testing in addition to ion chromatography. It also helped them to get instrument time scheduled so that there would be no disruption to the project. And if the quote had kind of been glossed over at this point and treated simply as a price tag, it could have led to some difficulties further down the road. Next thing I'd like to talk about is the sample submission form. So this is a really important piece of information that's going to help determine sample requirements and ultimately the protocol design of the experiment. So the first thing, or I suppose I should say, the things that you'll typically see in the sample submission form are going to be things to define the device, define the usage, request for instructions for use, schematics, materials of construction, processing materials, and potentially previous chemical characterization data. And so on this last one, we often get questions about this. And the reason that it's important is that this information can help in the data interpretation step, which we'll see later on can be fairly involved. And so that chemical context on this device or previous experiments and knowledge about it can help to make sure that the project stays on time and on budget. We do understand, though, sometimes that the components of the device or the raw materials may not have full characterization information. So it's not absolutely necessary to have that data, but every little bit can definitely help. And if a study sponsor has all of that collected and ready to share and shares it at the beginning, then the chemist performing the data interpretation can quickly look through it when they run into something where that would be helpful. So in the previous slide, I spoke briefly about determining the sample requirements, and I'd like to investigate that a little bit more detail. So what you see in the top left here is table 1 from part 12, and this is discussing the dose-based threshold for devices based on the patient exposure. Below that, you see a table with the various dose-based thresholds and some relevant parameters and device counts for a hypothetical device, which I've calculated just to illustrate the point. What's important to know, though, is that the Analytical Evaluation Threshold, or AET, is proportional to the dose-based threshold, and the range on that number is almost 2 orders of magnitude. So on a limited device, we're going from 120 micrograms per day down to a lifetime device exposure, where it's 1.5 micrograms per day. So again, that's almost 2 orders of magnitude, and the sample requirements can increase drastically. So for our hypothetical device, where we're doing a limited contact assessment, our dose-based threshold is 120. For this device that I've chosen with a surface area of 42 square centimeters, we can do 1 extraction at the recommended ratio, 14 mils. We don't need to do any concentration. We have a device count of 9. Moving to a prolonged study, though, we now need to do NVR to demonstrate exhaustion, so we double our device count. So we go from 9 to 18. Looking at the longer term exposures, we have dose-based thresholds of 20 and 10. So this does lower the AET, and this necessitates concentration prior to analysis on instruments to make sure that our AET is above the limit of quantitation for the characterization techniques. So luckily, we still only need 1 device, because that extraction volume of 14 mils is still sufficient to hit that AET target. But we do have to concentrate a 2-fold concentration and then a 4-fold for the dose-based threshold of 10. But we're still at 18 devices, 9 for our extractions and 9 for NVR. But when we go to 1.5 dose-based threshold, we have to concentrate 18-fold to be able to get our AT above the LOQ. And what this means is that 14 mil extraction volume is not going to be sufficient. So we have to extract more devices, and our extraction volume also increases. And so our device count reflects this. So it goes up to 54. So as you can see from the slide, being accurate on the device usage, the patient exposure in terms of the number of devices a patient can be exposed to, and that duration, that's all very important information to make sure that the sample requirements are accurate. You know, we understand that you know, devices can be very expensive. In fact, we've had projects where the devices used in the study were more expensive than the study itself. And we also know that with lean manufacturing, sometimes getting enough devices can be an issue, and they may even be manufactured specifically for it. So getting this information accurate and early is very helpful for giving an accurate estimation of sample requirements so that the project can be kept moving along. So I have another case study, and this is discussing the chemical context. This was a study that was prepared by a colleague of mine, Dr. Annalise Pfaff, who's a scientist here at EAG Laboratories. And she had a project where she was performing the LCMS annotation, and it was a polymeric product that underwent in situ polymerization. So it's a 2-part copolymer with a radical initiator. And what was interesting about this project was that the formula of both monomers contains the necessary elements to construct toxic compounds, that is, things in the cohort of concern, but those monomers in the initiator are not toxic. And so on the left was the first pass on the LCMS data. So we had numerous compounds that were above the AET, and the ones that contained copolymer were potential cohort of concern compounds. Now, if given sufficient time, Dr. Pfaff could hammer through these and ultimately determine that these did not belong with the cohort of concern, but it would have been a very involved project, and it would have definitely shot over the projected timeline. On the other hand, with this data, if she ran with it quickly and was not able to eliminate the possibility that these were cohort of concern compounds, she would generate a chemical characterization report, which really wouldn't lead to a favorable TRA. However, since the client was very forthcoming with the materials of construction and the process for the polymerization, she was able to leverage this data and quickly determine that all these potential cohort of concern compounds were actually initiator fragments and copolymer oligomers with initiator fragments. And so in this way, because the study sponsor provided all this information on the front end, she was able to quickly get this project turned around and generate a chemical characterization report that supported the device safety and led to a successful TRA within the budget and timeline originally projected. Another thing that we sometimes like to see is an example device. So we don't need this for every single project, but it can definitely be helpful for some of the ones where the extraction setup is a little bit more challenging, or that there may be handling or safety concerns with the device, right? So I'm thinking of drug-coated projects and maybe devices with trocars or sharps, other kind of puncture, sharp, hazard type things. So having this example device can be really helpful because there can be some brief practice in handling and setting up the extraction. I have an example down here below where we have a simulated device. It's actually just a serological pipette, but this is an approach we've used for some devices where we didn't want to extract the entire device. Since if we're provided that that example device, we can practice this setup and make sure that we can extract only the patient contacting portions rather than the entire device. So that can be really helpful. And even though this device, it's an additional device that costs extra money, we can identify problems in this step that could lead to the extractions failing, right? So you think about, I'm sacrificing one device on the front end, but that could save potentially the 9 devices that I'm using later on in the project. Can also do feasibility studies. So these are studies prior to the full study where we're looking at things like solvent compatibility, extraction setup, handling of unusual materials, and also some scouting of extractables. So these are all important because we can look at solvent compatibility, right? We can look at swelling and degradation and find solvents and demonstrate that they will not cause problems with the extraction on a single device. We can check that extraction setup, kind of like I alluded to in the last slide. We can also walk through the handling of unusual materials. Scouting extractables can help as well. That can help forecast if there's going to be any extension to the standard turnaround time. And all of this leads to cost and time savings. A feasibility study can be conducted pretty quickly. In a hurry, it can be about 5 business days. And so you can see where that could be very useful if you consume a limited number of devices in a feasibility study and determine important experimental parameters that could undermine an entire study. So again, it's another way that we can control the risk around cost and time overruns. So I have a case study about that I'd kind of like to discuss. So the image on the right is not the particular device that we were analyzing. This is actually a very early heart lung machine. But the device that we were looking at in terms of flow path was not much simpler than this. So we had this large device with a complicated flow path, and we had an exchange membrane with a large solvent capacity. We knew that the exchange membrane was incompatible with pure alcohols. So we had to answer some questions. What solvents, what volumes would we be able to use? What was the total internal volume? What would the filling process look like? And how much solvent would that exchange membrane retain? So all of these questions ended up being answered by sacrificing one device, which admittedly was expensive, in the range of tens of thousands of dollars. But if we hadn't done this part of it, we know for a fact that our initial approach would have failed and we would have burned 9 devices on the full study. So you can think about the difference between spending $10,000 on the front end versus $90,000 additional dollars when you have to redo the entire study, not to mention the effect on time. So again, this is something we've done practically in the past, and it's definitely worth considering for complicated devices in terms of keeping the project on target. So at this point, if all this work has been completed, we can then proceed with writing the protocol. It's going to describe the device preparation, extraction conditions, AT calculation, analysis parameters, reporting format. It can also address particulates that may or may not be seen. So this is helpful because it then fleshes out the timeline, and you also have a document that you can submit for pre-submission review or review by your toxicologist or internal team. So real quick, before we wrap it up, I'd just like to touch on particulates. There are expected particulates and there are unexpected particulates. A good protocol should address this, and if you know that there may be particulates, you should definitely bring it up in your study design to the analytical team as well as the toxicologist. There's questions about what to do with these. The standard says to isolate and characterize. That can be done, but the question is where. Some device manufacturers, they actually like to characterize them on site where they have their particle laboratories and libraries that they can leverage for identification and characterization. It can also be done at the lab doing the Part 18 testing, and it touches on the stability question, which should definitely be considered and addressed, right? Will the particles be stable through shipping? So I'd just like to wrap it up just by going back over briefly what we've discussed. So we discussed the non-disclosure agreement, the quote, sample submission form, and those initial preliminary communications to start designing our study. We then discussed, providing an example device, the benefits of feasibility studies, and then the protocol and what it should contain. So I think I've done a pretty good job of discussing all these things and putting it again in that context. And the idea that I don't think I could say enough is that the study sponsor, product manager, the analytical team, and tox team working together are going to generate a successful study. All right, so at this point, I'd like to hand it over to Dr. Batting.
Hi, everyone. I'm Melissa Batting. I am a consultant at Exponent, and I'm going to be talking about the importance of chemical characterization study planning with a toxicologist. And just for a brief background on myself, I am a toxicologist and I specialize in medical device biocompatibility and safety. So first, a little bit of background on Exponent. I realize most of you are probably familiar with Eurofins EAG, but maybe not with Exponent. So we are an engineering and scientific consulting firm that has been around for over 50 years, and we have a lot of expertise in a lot of different technical areas, and we have offices all across North America and some in Europe and Asia as well. So briefly, our expertise covers a number of technical areas, including health sciences. That's the practice that my colleague Megan and I are a part of, but we often collaborate with our colleagues in other practices, and especially when it comes to medical devices. We are often working with the biomedical engineering and sciences team, and then we have our own in-house polymer science specialists and materials chemistry scientists. So we are working with chemists inside and outside of Exponent. Now on to our specific chemical characterization expertise as toxicologists. If you've been a part of any of these studies in the past, you know that a toxicologist is needed after the chemical characterization study to assess the resulting extractables and leachables data for toxicological risks. However, we also work with test labs and study sponsors before the study is underway to help plan this extraction testing. I know Dr. Ferko touched a lot on, put a lot of plugs in for how toxicologists are helpful on this team. It really can be and should be a collaborative effort, because to best run these studies, which can be pretty challenging and complicated, we have to consider a lot of different aspects, including the device materials of construction, the chemistry methods, and of course, the toxicology. This schematic here just demonstrates the sort of general overview of how we combine our areas of expertise for chemical characterization and toxicological risk assessments. So the first part here, where you identify extraction conditions for your device, that is typically done by a collaborative effort between the chemistry team and toxicologists, if you're working with a toxicologist. And then there's the extraction procedure itself and the chemical analysis, where the leachables and extractables are identified and quantified. That's, of course, conducted by the chemistry team, and then the toxicological risk assessment is performed by the tox team. And like I said, it's useful to include a toxicologist before your testing, and here are some of the considerations that we should and can be involved in. So one part is test article selection. We always ask, is the device in its final finished form? Of course, that's the ideal and that's the goal, but it's not always feasible. So we can help to select and justify the use of coupons or test articles that are not final. We're also helping with a lot of times making the decision of whether a test article can be or should be cut into smaller pieces to help with the extraction ratios. Sometimes a component of the device needs to be removed before extractions or assembled prior to extractions to best represent actual patient contacting surfaces for extraction. We also help a lot on decision-making for the extraction conditions. Of course, the lab is going to propose those. FDA or other agencies may suggest them, and the ISO 10993 standards propose and recommend them, but there are a lot of nuances to that, right? So is it an exhaustive extraction study? Are you trying to get more at a simulated use more clinically relevant, leachables test. That's going to impact the temperatures, the solvents, and other parameters that are selected for the extraction conditions. And if you're deviating from standard temperature, time, solvents from what's used typically, you often need to provide justification, whether that's ahead of time to the agency that you're dealing with or on the back end if changes need to be made. And as toxicologists, we can certainly help with this. Other considerations to think about before testing is interpretation and best implementation of feedback from regulatory reviewers. So it's not always clear from reviewer feedback how it may relate to your specific device. For example, I recently saw feedback requesting the standard 50 degrees Celsius extraction temperature, but for an externally communicating device that would never get near 50 degrees Celsius, used at ambient room temperatures. So we pushed back on that. We provided a literature-based rationale for why a 37 degree Celsius extraction temperature would be appropriate and still worst case for an extraction scenario. That was considered acceptable by the reviewers, and I'm really glad that we asked about that so that we don't overdo it with our extractions. We also help with reviewing proposals and protocols. Dr. Ferko talked about this. So the proposal is, of course, a first look at what the lab proposes to do. And we want to make sure that the methods address not only what's needed for the chemical characterization study, but will these methods be suitable for the regulatory submission, especially if there's already feedback in play. I always look for things like sample manipulation. So to meet those rigorously low and sensitive reporting thresholds, the AETs, we are in a situation where oftentimes the chemistry team has to concentrate samples and apply dilution factors. So while that makes sense from a technical standpoint, that is a comment that usually comes up from reviewers on the regulatory side. So I always want to know if the lab has appropriate spike and recovery data to support sample manipulation. I also look at reference standards for each analytical method to make sure that those are appropriate. And I always want to discuss resulting data output. So we need for the tox risk assessment, we need the results to be on a mass per device basis or mass per test article. So we want to make sure that we're aligned with the test lab on that. One of the last considerations that I wanted to discuss about before testing is the derivation of the Analytical Evaluation Threshold, the AET. So a lot of times the lab provides this as part of their protocol, but as a toxicologist, we want to take a look and make sure that all of the factors that go into this formula are correct and that we agree with them from a toxicological standpoint. So one thing is the maximum number of devices per patient per day. It's important, of course, to consider the maximum number of devices that one could contact during use. And of course, the duration of patient contact with the device is really important. It sometimes surprises me how often it's overlooked that there is the potential for cumulative exposure when devices are used short term, even less than 1 day. But if there's potential for cumulative exposure, we need to consider that as part of the AET calculation. And of course, the dose-based threshold, we need to make sure that that's appropriate. That uses a TTC or threshold of toxicological concern value. So we want to make sure that's right from the beginning before the study's kicked off, because that can have real implications for the acceptability of the toxicological risk assessment to the regulatory reviewers. So now I'm going to discuss some of the considerations during testing that a toxicological expert can be involved in. So sometimes things go really smoothly, but oftentimes there are issues or challenges that can arise during chemical characterization testing. I have some of them listed here. For example, solvent incompatibility. I know a lot of labs do solvent compatibility testing at the start now, like Philip talked about, but that doesn't necessarily catch everything. Sometimes during those iterative, exhaustive extractions, the device can degrade or fall apart. You can have really, really high non-volatile residue or NVR masses that indicate that there's some degradation or some issues happening with compatibility of the solvents. And sometimes that can lead to anticipated complex and very large extractables profiles. That's something that we want to be made aware of during the testing. And of course, any deviations from the ISO 10993 Part 18 standard or from the test lab protocol needs to be brought to the attention of the toxicologist so that we can plan for that. And you know, in the second bullet point here, we help the study sponsor often to make these decisions that could impact the tox risk assessment. So really important to keep us involved. And we're able to better prepare for a successful tox risk assessment if we are kept aware of these issues as they occur. And I want to go over, it sounds like I've already talked about benefits, but just to sort of bring this home, the benefits of including a toxicologist in your chemical characterization study planning include a number of things. First, like I alluded to earlier, we can help best incorporate agency feedback. We want to ensure that that regulatory input has been adequately addressed in the study design. And a lot of times we can discuss options with the test lab, including, is this even feasible for certain requests from regulatory reviewers? I'm working on a project right now, actually with chemists at Eurofins EAG, where the agency has asked for gas pathway testing for volatile organic compounds, VOCs, via the standard ISO 18562. And specifically, they want it for a known additive that's applied to the outer surface of the device. However, I've done my research on it. The chemical's not volatile and it's not expected to leave the surface of the device. It's a very large molecular weight, non-volatile compound. So we're working with the chemists to come up with a way to provide technical data. You know, is this even feasible for one to conduct that type of testing based on the physiochemical characteristics of the additive in question? And is it even possible for the instrument to measure that type of compound? So this type of back and forth collaborative relationship is, like I said, really important, and we need to put together something to respond to this feedback. So a lot of times that involves both a tox and a chemistry perspective. And of course, the goal is to have a lower risk for agency deficiencies and the possible need to repeat studies. No one wants to be in that situation. And we're really familiar with ISO 10993 Part 18, the standard for chemical characterization, along with ISO 10993 Part 17, which is the standard for tox risk assessments of medical device constituents. So me and my team, we know the analytical chemistry, chemical characterization standard, and the tox risk assessment standard. It's important to know how to best comply with them. And this last little bullet here, I think it's really important also to provide insight on current and emerging topics and areas of concern and opinions from the agency reviewers that may be not part of the ISO standards, but they come up in deficiencies and we see them over and over again. And a lot of times it has to do with the interpretation of certain clauses or approaches. So we are well versed in those nuanced opinions, and we can help with that. We also provide input on decisions for any challenges or issues that arise during testing, as I mentioned earlier, keeping in mind how any deviations and specific outcomes could impact the tox risk assessment, which of course we want to ultimately be favorable. And as I said before, I think it's really important to establish a relationship with the chemistry team and the test lab, because we really can have a collaborative effort with not only the planning, but also the data analysis and review phase of the study. As my colleague Megan will be discussing after the first Q&A session, the toxicologist sometimes needs additional information from the chemists to properly conduct the risk assessment, for example, with unknowns or poorly characterized or poorly identified compounds. And so it's great to have that relationship already established. And now we're moving on to our first Q&A session, which is going to be moderated by Sahil.
Thanks, Melissa and Philip, for the insightful presentations. Now we're going to have all of our presenters join for a panel discussion. And wow, we do have quite a few questions from the audience here, so I'll do my best to pick a couple here. And The first question, we'll go ahead and get started, is what is the best way to go about initiating a program? Who do I reach out to and what should I consider? So maybe Philip, do you want to start with that?
Sure. So I think the best thing for initiating your program in terms of who to reach out to, you can reach out to a toxicologist or organization, toxicology organization that you know and you worked with previously, or maybe have a reference for, or you can reach out to the lab. The important thing is though is that whichever you start with, you want to kind of make sure that the other parts also getting lined up. Maybe you have both and you need to put them in touch with each other, or you can ask for recommendations. I think the other part though, and I touched on this in my talk initially, is that's the time to go ahead and start collecting all that information, right? You may have to reach out to your vendors or go looking for old chemical characterization reports, that kind of stuff. If you start that literature gathering, the more of that you have ready to go at the beginning, the better it'll be.
And Melissa, do you have anything to add? I'm pretty sure you get also requests for how to start a chemical characterization. So
yeah, we do. It totally depends. It either comes in through our clients who know they want to go with us for help on the toxicology side and they want help identifying solid labs, or like Philip said, they know the lab or they come to the lab and then will ultimately need help finding a toxicologist. So either way is a great way to start.
Okay, very good. We'll move on to the next question. The next question I have is, what do I need to do as a study sponsor to set myself up for success, keeping timelines and cost in mind?
Yeah, I'll start. So, I think I sort of answered this one already, but we can go in a little more detail. Again, from the laboratory side, it's getting all that supporting information that you have around gathered in one place, right? You may have to request it from, other stakeholders in your organization. You may have to go to a vendor or vendors to collect all that. And some of it could be things that you don't necessarily consider, right? Packaging is important. Processing aids, like from the cleaning process, those are important. Things that may be exposed to in the manufacturing process. All that kind of information can be very helpful and keep things on time when it comes to the interpretation of the data.
I'll chime in from the tox side since we're always the ones waiting on the data. Most of the time, this is the last part or one of the last parts of a biocompatibility evaluation. I always encourage study sponsors and our clients to get started on chemical characterization as soon as you can, because things happen and it can take longer. You may have some compounds that are concerning that fail a risk assessment, and then you're stuck. And you may need to then run a simulated use leachables test or something, or a targeted test for specific chemicals, and then you don't have time to successfully do that. So I always encourage, if you have 3 months left until your submission deadline, do not start your chemical characterization test then. You should start it 6 months earlier, if possible. So that's my advice for success, is giving the experts, the chemists and the toxicologists, the proper time.
Yeah, that's great feedback, Melissa. And that kind of dives into another question we have from the audience in terms of, well, the question is chemical characterization needed for a water-soluble device? But I've also seen some questions centered around, how do I know if chemical characterization is provided or needed? So maybe you can give some insight on, certain situations and devices that chemical characterization is required.
Yes, I think anytime you're trying to cover the biocompatibility endpoints that are long-term and systemic. So if you have a device with prolonged or permanent contact, again, considering cumulative exposures, repeated exposures, then you should consider that you're probably going to have to do chemical characterization. I've seen requests lately from FDA reviewers, even for limited contact, short-term contact, even surface contacting devices. So it seems to be getting a little bit more stringent with what regulatory reviewers expect. but ultimately the tox risk assessment is supposed to address those long-term systemic tox endpoints. So we think any device that's potentially going to have an impact on safety long-term.
Yeah, great. That's an excellent answer, Melissa. Philip, do you have anything to add there?
You know, I think the question about the water soluble device, I think it's, you know, it's a great question from the chemical side as well, right? What do we do with these devices that may be designed to dissolve or be resorbed or these sorts of things? And that's why, you know, it's important to have, you know, it can be, you know, it may be things, you know, even just physical property information from the manufacturer of a polymer material. Having that can be helpful, and then a feasibility study, right, to figure out, okay, how are we going to best meet the requirement that we've been given, right? The FDA says you need to do it, then you've got to do it. You can't just say it's too hard, right? You've got to do something, and so that's where the feasibility studies, I think, come in.
Yeah, great. That's an excellent point. The upfront process is a lot of times overlooked in these studies. So, yeah, well, great answers. I appreciate that, Melissa and Philip. Just to keep everything on track, we're going to go ahead and move on to the next set of our presentations that will focus on study completion and results with Dr. Ferko again and Dr. Arnold. And then we'll have another question and answer session followed shortly after that. So I will turn it back over to Dr. Ferko.
All right. Thank you, Sil. Okay, so in the first part, I discussed all those steps leading up to the initiation of a chemical characterization study. So at this point, we're now going to talk about executing the study, right? We have the protocol. It's been reviewed and approved by all the appropriate groups. And so now we're actually ready to step into the lab and start working. So the approach that I'm going to use in this talk is to go through the steps chronologically, discuss what's going on in those steps, maybe challenges or ways that they can increase or decrease, and then also, you know, what's an average time in business days to completion of that step. So before we get started on that, though, I have to say I'm picking a kind of a representative project, right? So we've got a reasonable extractables profile, right? It's not there are things above the AET. There's going to be some work to be done, but it's not a particularly complicated extractables profile. That can definitely have an influence. We're also assuming that we're going to do 3 extractions, right? This is an exhaustive device, but we're not having challenging issues like, you know, continuing NVR where it doesn't seem to be tapering off much. It's not a complicated material that may be degrading. So fairly simple that way. Those are the fringe cases. Now I'll discuss those a little bit towards the end, but in general, in your study, the first thing that's going to happen is the extraction, right? You have to prepare the device and perform your extractions. And this takes, for 3 cycles, it'll take about 10 business days. After that, there's the instrumental and gravimetric analysis. This usually takes about 3 business days. The next step is the data interpretation, and this can take about 25 business days. And this, in my opinion, represents the most hands-on time from the chemists in getting from the start of the project to the end of the project. This is also where we can see variability in the overall timeline of the project. And so there's a lot of visibility around the data interpretation and what is its impact going to be on the overall timeline. There's report compilation and review. So that's going on in parallel with the project as much as possible, but inevitably at the end, there's that last bit of compiling, the last bit and reviewing those final parts. And what we end up with is a project that runs about 40 business days. So on the extraction, this is going to take about 10 business days, and this includes device preparation time as well as the extraction time. So we're going to be performing a series of extraction cycles because this is an exhaustive device. And for each of those, we have to prepare the extract, run it, and then we're going to decant the extract and then refresh the extraction vehicle. So we're taking starting and finishing weights to make sure that we're not losing solvent. We're taking physical observations and recording those, maybe taking images as well to show, the device before and after extraction. So we can see if there's any visible change in the device. We can look at things like changes to the surface, or we can look at generation of particulates, can look at degradation, all these types of things that we need to document that, yes, we're looking at those and they're included in that report. We're also monitoring the temperature and extraction conditions to make sure that it is being performed as written in the protocol, that we've immersed the device, it's under continuous mechanical agitation, and the temperature is where it's supposed to be. So we're also going to be determining the non-volatile residue roughly in parallel. So you can see there's a lot going on around this. It's not, particularly challenging in any one thing, but we have to be very diligent in making sure that we record all these observations because they can become very important later on down the road when we're looking at the full story. So for extraction vehicle preparation, we've decanted these extracts from the devices, and we now need to prepare them for analysis. So in the case of the non-volatile residue samples, it's pretty simple. We're going to dry those completely, right? And that's why it's non-volatile, right? The volatile portion's removed, and so now we have something that we can analyze gravimetrically. Analytical samples are a little bit trickier. We're going to concentrate those. We're not going to completely dry them. If we completely dry them, there's concerns about things precipitating out that then don't go back into solution. So we've got to really watch carefully, make sure that we concentrate at the appropriate amount, but not dry it. So that can also involve, after the concentration, some dilution into appropriate solvents. For example, GC requires a portion of an organic solvent for analysis, and so we may dilute it in an alcohol to make it more appropriate for analysis by the instrument. ICP-MS needs an aqueous acidic diluent. So there'll be some sample handling here, and the purpose of this is to render the samples compatible with the instruments that they're going to be analyzed by and to ensure the quality of the data. So let's talk a little bit more about the non-volatile residue analysis. The purpose of the non-volatile residue analysis is to determine exhaustive extraction in this study, right? So what we're looking to do is to show that, the last extract that's going to be analyzed, we have less than 10% of the non-volatile residue that was observed in the first one. That's the definition in the standard of exhaustion. So there's two approaches that we can use. We can use a gravimetric method or we can use a chromatographic method. The previous thing I alluded to is really the gravimetric method, right? So we take the sample, we dry it completely in a tiered container, and then we simply measure the mass. What's nice about doing a gravimetric measurement is that, a balance in a sense is a universal detector, right? If you have matter and it's enough that it's a mass that can be observed by the balance, it will be observed, right? And so this is kind of an orthogonal approach to the instrumental techniques. Some instances do exist, though, where gravimetric method isn't really suitable. So think of those devices that are made out of something like stainless steel or titanium, some very impervious material of construction that doesn't really leach or degrade under remotely normal circumstances, right? So for those, we may not get much off, and it may not be enough to be able to observe by a balance. And that's where we can then go to chromatographic methods. So we lose that orthogonal viewpoint, but at the same time, we can see much, much lower in terms of the detection limits by the chromatographic methods. So we can also demonstrate exhaustion that way. So if we do have gravimetric data, though, we do like to compare the gravimetric NVR results to the chromatographic results, regardless of if we're demonstrating exhaustion by the gravimetric method. So this exhaustion to NVR analytical comparison is requested by agencies, and there's two fringe cases, right? The best is you're close, right? And close as far as people are currently concerned, it's usually within an order of magnitude. It may seem large, but we're talking about pretty small amounts here, for some of the mass balance stuff on the gravimetric side. So that's kind of the position that things are at currently. But if our NVR is significantly less than the summation of our analytical recoveries, we're encouraged to examine our methods, right? And most labs are already doing this. They're making changes to make sure that their analytical recoveries are reasonable. One good example of this is using surrogate standards to more accurately semi-quantitate what's being detected. The other case, though, is where our non-volatile residue is significantly higher than our analytical recoveries. And at that point, we really should be examining our residue. So we can do that by a variety of methods. Probably the go-to one, and the one that you'll see done routinely, is FTIR spectroscopy. And this is good for getting kind of a bulk analysis of the material. And you can get some structural information on what you're observing. So some common things that you'll see there are polymeric species, right? So if you have a polymeric medical device and you have degradation, you may have pretty large oligomers, and you can then see these by FTIR and demonstrate that that's really what you're that's what the bulk of that NVR is, and that's what's pushing it up above the analytical recoveries. But there are other methods that can be used as well, depending on what kind of material you're looking at and how much detail you want to go into. Two examples are ion chromatography, which I discussed previously. Another is gel permeation chromatography, which is another way to look at polymers but gives size distribution information. So next, I'd like to talk about the instrumental analysis. So we've got our samples. We've taken them, we've concentrated them, we've rendered them suitable for the instruments they're going to be run on. And then in a standard study, we would run them through these listed techniques here: heated headspace, GC, direct inject, LCMS, ICP-MS. Others can be added, right? We talked about that before. But the purpose of this is to, in terms of molecular weight, the polarity, volatility, boiling point, other characteristics, right? So that we're really looking at this from every angle to spot all the things that are being extracted out of our device. But the other part is that not only do we want to get instrument responses for all these so that we can do the semi-quantitation, we also need to generate data sufficient for identification of analytes. And that's why if you look at these, you'll notice that chemical characterization is mass spectrometry heavy. That's really important because that provides us the data to identify these compounds and give that structural information that's necessary for the toxicologist to complete the TRA. So we've talked a couple times about the AET, right? I mentioned it in the discussion of the protocol, and Melissa touched on it as well, and making sure that it's appropriate to the device. So now that we've made sure it's appropriate and we've done the preliminary calculations, now we need to examine it and apply it specifically to our instrumental techniques, right? So we've already calculated it, but when we calculated it as a concentration in solution, right? Now we need to convert it to an area threshold. Since our quantitation is based off of peak area and these chromatographic methods, we want to know above what area do we need to analyze a peak. And if it's below that area, it's below the AT, we don't have to worry about it. And so the area thresholds will be calculated based off the calibration curves generated at the time of analysis. And what we'll do is we then go through and we count all the peaks that are above the AT and neglect the ones below. This is really important. It gives us a peak count. And a peak count is a measure of the work to be done on the project from here to the completion of that chemical characterization report. So this is the point where we can revise the timeline and where things get to be really accurate. Now, I don't want to suggest to anyone that there's no way to predict how long the study will take prior to this. It's just that this is the point where it really firms up. Certain devices we can look at and we know it's probably not going to be a large peak count. A great example of this would be, again, those stainless steel surgical devices, right? Very high AET, very little extracting off. We're going to be in the clear, almost certainly. On the other hand, we can look at a device, something very large and complicated or made of an unusual material, and you may have data already that there may be degradation issues or some other aspect of it that's going to make for a very complicated extractables profile, and we can predict ahead of time that yes, it may take longer. So those conversations are important, but again, peak count here tells us what's left to be done. I want to talk a little bit on the data interpretation, really about the quantity of data and the handling process to help people understand why it takes 25 days. It may seem like a lot of time, but I think once I walk you through the slide, I think you'll understand that 25 days is actually really impressive for what has to be done. So a lot of mass spectrometry techniques used in chemical characterization are now moving to high resolution instruments. And with these instruments and the fast cycle speeds on them, we're looking at gigabytes of data per measurement with hundreds of measurements per technique per project. So this is a very large amount of data. So on the right, I have an example here. I have a total ion chromatogram from a single injection from a project. So it'd be one of hundreds of injections done for this project on this instrumental technique. At time instances throughout this total ion chromatogram, we're collecting MS spectra as shown below. So in the instance of, say, LC, just as an example, we're collecting about 7,000 MS spectra per injection. So think of it as, you know, a book of 700 pages of images that have to be evaluated. So that works out with over 100 injections. That's 100 books. It's 7,000 pages per book. So we're creeping up on close to 1 million images or pages of a book that have to be evaluated. And that's per instrumental technique, right? We have 3 chromatographic methods. So we're looking at about 3 million pages, right? That's a lot. So getting through it in 25 days, 25 business days is actually an accomplishment. And part of what makes this possible is leveraging tools that automate alignment, deconvolution, and data interpretation to the limits possible. I emphasize the limits because there are definitely limits. We cannot hand this over to computers and just let them do it. If you build a simple system like that, you will make simple mistakes, I assure you. So what we can do though on that though is we can at least be looking at MSN libraries. This is an automated process, at least on the first pass. So NIST or other commercially available libraries can be used and comparisons can be made in an automated fashion. You can also generate internal libraries, which is really important because these can be targeted towards the types of compounds you more typically see in medical devices. But I want to reinforce that review by a subject matter expert is still essential to produce reliable data. You cannot simply take the highest match factor compound and run with that. You will end up generating data that's not really accurate and will not support a TRA. So that review step is really important, and that's a lot of what's going on in this 25 business day section. It's the chemist looking at the data, making sure it makes sense, making sure it agrees with itself, making sure it agrees with the other techniques, and any information that the study sponsor can provide, right? Materials of construction, previous characterization work, that sort of thing. So after getting over the hump of the data interpretation, we're wrapping up report assembly. This takes about 2 business days. So we're assembling a multi-technique report, and we're finishing up the technical review and the correlation of the data, right? For the most part, we're trying to do that as we go, as we generate the data and interpret it, so that we're not doing it completely in serial, but we can't fully parallelize it. There does need to be a little bit more at the end, right? We'll also be making the last reviews to make sure we're meeting our reporting requirements, looking back at the protocol, any other discussions that were had around the report format. And the whole idea is that we generate a nice comprehensive packaged report that's going to give the toxicologist the information necessary to perform that TRA. That's the goal. The goal is we produce the report, it stands on its own, and the toxicologist can take it and make the assessment and be done. It doesn't always happen that way, but that's definitely the goal, and we're always striving towards it. But the other part is the lab then is going to stand by to assist with additional information. So Melissa alluded to that earlier about, you know, sometimes you have to go back to the chemists and ask additional questions, and so you've got to be ready to do that. And that certainly is something important that's part of the lab's responsibility, in my opinion, to continue supporting these studies to get them over the finish line. So that's kind of the summary of our standard project, you know, our exhaustive 3-cycle one. So I'd like to talk just a little bit about sort of what makes it into a fringe case, right? The easy case initially, you know, is going to be your exaggerated ones, right? We've got a high dose base threshold, 1 cycle of extractions. There's a good chance that we will see very little or nothing above that high dose base threshold, especially if it's a simple device, right? Stainless steel, we see a little bit of surfactant. We've seen those a million times. We've got them in our library, the internal one, so it's easy, right? We can knock it out. We can turn that around very quickly. Data interpretation can go down to single-digit days easily, right? So that's one way that we can pull it forward, but there's the other side, right? It can go longer. And these are going to be complicated devices or materials, things where we have larger number of extraction cycles, or we end up with extended data interpretation. Those two things are related. They're not completely orthogonal, but they're not necessarily the same thing. I'll share just a brief story about a project that we did have where we had a device and it was fairly large. It was a polymeric material. There's a decent amount of polymer additives in it. And the number of extraction cycles ran on for quite a while. I believe it ended up going 13 cycles before, in the end, the decision was not made that it was truly exhaustive. It was that we needed to justify why we were stopping at that point. And so those kind of projects can be spotted ahead of time. So if everyone's being open with each other about it, they could say this one's probably going to take longer than the standard time, but it can happen. It can, you know, sometimes be a surprise. But what's important is that the lab's communicating with the study sponsor and the toxicologist, so that as soon as possible, everyone knows and can be ready to deal with that change, right? The other part is extended data interpretation. If you have a fairly complicated extractable profile, it can take longer to hash through that data. But again, those materials can kind of be spotted ahead of time. Biologically derived materials are probably a really good example of that. Those are just naturally more complicated, and so they tend to take longer, and you can know that looking at the materials of construction that it may take longer. So again, just a quick recap. We've got our steps on the left, our extraction, which for 3 cycles, about 10 business days. Instrumental and gravimetric analysis adds an additional 3 business days, pretty quick. The data interpretation is the bulk of the study, 25 business days. Last little few days at the end for the report compilation and review, and that gives us a total average time of around 40 business days. All right, so at this point, I would like to hand things off to Megan.
All right. Okay. Hi, everyone. Thank you for joining. Today, my talk is going to focus on how we toxicologists work to address findings from the chemical characterization studies, as well as how we present your data in the best light for your regulatory submission. So as Drs. Batting and Ferko discussed, having a good working relationship with your toxicologists and chemists is incredibly beneficial for planning your chemical characterization study. However, this working relationship continues to be critical once the study has been completed. So for instance, in order to meet regulatory deadlines, we often begin our risk assessments with draft data from the chemical characterization reports. And because we're initially working off of draft data, and because very different compounds can have similar peaks, it's not uncommon for toxicologists to ask for clarification on these compound identities. So for example, in the past, we've had projects where we've had to ask the chemists to clarify whether an antioxidant stabilizer, such as vitamin E, was misidentified as a steroid, because we would expect to see something like an antioxidant stabilizer from a polyethylene device, whereas we wouldn't expect to see a steroid. And once we've gone back and forth on the chemical identities and gotten everything finalized, we can then start our toxicological risk assessment. And so as we're going through the TRAs, one inevitable outcome is that we're going to have to address constituents that don't have toxicity data. So in order to address these compounds, we review the entire data set and determine whether any other constituents could be grouped together based on similarity, such as physicochemical properties, functional chemical class, or predicted toxicological endpoints. And if those other constituents have toxicity data, we can then rely on those group members to address the group as a whole, including those without data. So for example, when we have multiple phthalates, such as those shown on the slide, in the data set, we can rely on toxicity data for the one that does have data, or for the worst case category member if multiple of the constituents have data, in order to address this group of phthalates in one TRA. In addition, we can also group fragments of larger molecules and rely on the toxicity data from the parent compound to address the toxicity of the metabolite, or vice versa, depending on if the metabolite has data but not the parent compound. That said, it's not always possible to form a group with other constituents, or sometimes we encounter that none of the grouped constituents have toxicity data. So in these situations, we rely on our expertise and knowledge of chemical classes, as well as the use of softwares like OECD QSAR Toolbox to identify suitable analogs that do have toxicity data. And so once we identify a suitable surrogate for toxicity read across, we always make sure to provide a robust justification for our selection. That'll include things like functional groups, that's in the constituent and surrogates. So they are all aldehydes, whether or not they belong to a specific chemical category or class. So all low molecular weight siloxanes together versus the larger ones. We make sure to address things like incremental change across the categories. So shorter carbon chain length alkanes have increased bioavailability than longer chain length alkanes. So we always make sure to rely on the shorter ones when conducting our TRA to address the longer ones. We also make sure to address, when relevant, precursors or breakdown products for the constituent. So one example is Ergonox 1010. This is an antioxidant stabilizer commonly used in polyethylene devices. And in the extraction studies for these devices, we often get a lot of ditert butyl compounds that look similar to butylated hydroxytoluene, or BHT. So even though the identified constituents are likely fragments of Ergonox 1010, to be extra conservative, we'll rely on toxicity data for the more potent compound BHT for read across. But in our justification, we can provide additional context in the TRA that this surrogate selection is likely overly conservative and that the constituents are likely fragments of the Ergonox 1010. Thus far, I've focused on how we can address individual or groups of constituents, but sometimes the unexpected outcome is the data set itself. So the number of constituents in an untargeted exhaustive extraction study can vary widely from one device material to another. So we know to expect more constituents that need to be addressed from peak devices, lower hundreds, versus those from a titanium device where there might be just a handful of manufacturing residuals. But sometimes we get unexpectedly large data sets where we have to deal with hundreds more compounds than we originally expected, and these still need to be addressed within those hard regulatory deadlines. So we have a couple tools in our toolbox in order to get this accomplished and meet the deadline. The first thing we can do is group compounds, as I previously described. We can also use the new toxicological screening limit from ISO 10993 Part 17, which we all know and love, but we all know that we can't always use the TSL. If we're dealing with a medical device, for example, that's used with neonates, we can't apply that TSL. So we have some backup screening methods that we like to rely on when appropriate, such as the TTC-based mutagenicity screener, where we go through the constituents and determine which are not genotoxic. And this is important because we can then rely on higher TTC values. So for long-term permanent implants, in order to protect against carcinogenicity, that TTC threshold is 1.5 micrograms per day. But for the compounds we demonstrate to be non-genotoxic, we can then rely on 90 micrograms per day. So any constituents that are non-genotoxic and below that 90 micrograms per day threshold, we can then justify not requiring a full TRA. However, the success of this approach can be limited by the device material and the expected extractables profile. So we always make sure to consider this before going through this approach, as it can require a bit of effort on the front end. In addition, toxicologists, we also help put the results of the TRA into context for regulatory agencies. So we can address toxicologically acceptable, albeit low margins of safety by highlighting the clinical significance of the estimated worst case exposure, conservative assumptions used in the risk assessment, as well as the exaggerated extraction conditions that the chemical characterization study used. And sometimes when there's the presence of a large number of constituents that can be extracted at high concentrations, this can produce an unfavorable TRA, even when we're dealing with compounds and chemical classes of low toxicological concern like alkanes. So what we can do is provide justification that in this specific situation, the unfavorable risk assessment could be due to a solvent compatibility issue. And this can arise even when there's no evidence of degradation or solvent incompatibility conducted by the chemist prior to the extraction study. So when we encounter this, we like to highlight the conservative assumptions using the TRA and the clinical significance of the exaggerated extraction conditions. We can also provide this rationale as justification for a lack of solvent compatibility and not actual toxicological risk from the device. It's also not uncommon for us to encounter red flag compounds, so things like carcinogens, cohort of concern members, and compounds of high regulatory concern and in the device extracts. And we know when we get these red flag compounds that it can be unsettling for clients. But you can rest assured that the mere presence of these in your extracts does not guarantee an unfavorable outcome for your risk assessment. So we have extensive experience and rely on agency-specific guidance, as well as our published standards, which we love the new ISO 10993 Part 17 for actually including carcinogenicity risk assessment guidance, so that way we can assess the clinical risk to patients for those carcinogens, as well as cohort of concern members. Now, for compounds of high regulatory concern, these chemical classes are highly scrutinized by agencies, but there's not necessarily agency-specific guidance or published standards on the best practices for evaluating these compounds. And so addressing these compounds in the TRA can require extra care, especially when there are no toxicity data for the identified constituent. So when we're dealing with a constituent that does not have toxicity data and belongs to this type of chemical class, we always address whether the worst case class member, so for perfluorinateds, PFOA, for phthalates, DEHP, or for bisphenols, BPA, if these worst case class members are the most appropriate surrogates for toxicity read across. And if we've demonstrated that they're not the most appropriate, what we then do is identify a suitable surrogate and provide a robust justification for this decision. The acceptance of this justification is incredibly important for the acceptance of your TRA, especially because regulatory agencies like FDA, MDR, or Health Canada can rely on different acceptable exposure limits for the same chemical or chemical class, especially for those worst case class members. And so choosing the correct surrogate can make a lot of difference across agencies, whether or not your TRA will be considered acceptable. And so at Exponent, we rely on our knowledge of the agency-specific guidances that are published, as well as the outcomes of our frequent interactions with these agencies in order to best tailor the justifications we use in the TRA for the specific constituents in your device. And so your toxicologist regulatory expertise can be a huge asset in order to decrease the risk of deficiencies related to your TRA. And now I will turn it over to Sahil.
Thank you, Megan and Philip again. That was great. Now I'm going to ask for all of our presenters to join us again for another panel discussion. So we had another good amount of questions come in, so I'm going to try to pick and choose here. I think a good one to start with is how do you design a chemical characterization study to meet the new Part 17? So maybe I'll start with there, Megan, and we can kind of go back and forth there.
Yeah, so for the new Part 17, that primarily affects what we do as toxicologists on the back end of the study. So once we have the data and how we conduct the TRA, but as far as the extraction conditions and test article preparation, all of that's outlined in ISO 10993 Part 18, and that isn't changed by the new Part 17.
Okay. So then the next one, I'm sure you're gonna enjoy answering this one. The next question is, how do you manage the presence of unknown compounds in an ENL study from a toxicological perspective? And I'll go ahead and address that to you again, Megan, to start off there.
Yeah, so this is definitely where having that good working relationship with your chemist comes into play, and why we love working with Eurofins so much. So in the new Part 17, we cannot use the TSL when we have unknown compounds, but if we have something like a chemical class or a partial identification. So let's say it's something like alkanes with a carbon chain length greater than 30. That's enough for us to be able to then, do our risk assessment, and we've identified it, clearly enough that we can use the TSL as well. So yeah, definitely having that relationship is great.
Okay.
And I'll chime in that sometimes we have these poorly identified compounds where the chemist is really able to help us, where we can say, looking at the device materials of construction, any additives, manufacturing process agents, anything like that, we can sort of suggest, do we think this is maybe related to this or that? And so yeah, we're usually able to work with the chemist to come up with some type of help, even if it's a partial structure.
Yep. Philip, anything to add there?
Yeah, although the question, it's about the toxicological perspective, I think it's worth mentioning that, with really with the newer equipment, with high resolution data and the, the detail that you can get from that, along with, device information, you can get something. Because if you think about it, if you're detecting these things by mass spectrometry, you can't detect something unless you're getting some sort of structural information, right? And I think that's where the interpretation part's important, and that's why you can't just have a computer churn out the results. You've got to have the subject matter experts involved, and it's good to have a diverse team. I'll say that, because we've got, in our laboratory, we have people that have industry manufacturing, chemistry experience, polymer experience, inorganic and organic, and having that kind of a group to kick a problem around, you can get something, figure something out, and be able to help out the toxicologist.
Okay, great answers. It looks like we have a little bit more time, so there is a question that came in around, are there any general precautions or things to consider for devices that contain absorbents? So maybe we'll start with you, Philip, on that.
Sure. So it's really important here that before you start the study, the full study, you have to know how is that going to play out when you go to remove your solvent, right? A lot of times when you have something that's adsorbent, like a filter or something that'll retain solvent, typically you're doing a fill on the device, right? And depending on the size of it, you say, okay, I'm putting in 10 mils, right? And if 10 mils is just enough to complete your study, but you decant it and you only get 5 out, that's a problem, right? You don't have enough solvent to even analyze, right? So you've got to figure out what's going to give you enough to complete your analysis, but then you also need to determine how are you going to address, the concentration, right? Because, for example, with the non-volatile residue, right? You decanted 5, technically that's only half of what was in that device, right? And are you going to consider it to all be in equilibrium and to be truly representative? Or, will you do additional extractions to support that, right? And that needs considered on the front end.
Great. And yeah, just to add to that, that kind of ties into the first part of the talk about, making sure you have an effective plan on the upfront process, solvent absorptivity, solvent compatibility. So great answer, Philip. Melissa, Megan, anything to add from your point of view from dealing with materials with absorbents?
No.
Okay. All right. Well, I want to thank you, Melissa, Megan, and Philip for the wonderful discussions today. It's been a pleasure to work with you guys on this. And I also want to thank everybody that joined, and I hope that you were able to learn something from this presentation. If there's anything that you want to dive into deeper, feel free to reach out to any of us with questions. I believe we're going to be sharing this presentation and also our contact information after the presentation. So thank you, everybody. And now I will turn it over to Melissa.
Thank you to our presenters and you for attending today's event. If we didn't get to your question today, we will follow up with you personally very soon. And once you leave today's event, you'll receive a survey on the presentations, and we would really appreciate your feedback. And so on behalf of Exponent and EAG Laboratories, Thank you for joining us and have a great afternoon.